Thank you everybody for tuning in for the recording of my talk today titled Education and Socioeconomic Status Mask the Identification of Brain Cell Types in GWAS of Mental Health and Disease. I'll start by talking a little bit about some differences between Mendelian phenotypes and complex traits, which is the topic of the presentation today. Mendelian phenotypes, as we see in the schematic on the top left of the screen there, are typically the result of a single causal gene, whereby you confer pretty much complete risk for the phenotype given a certain variant at that causal gene. Occasionally, there is an additional modifying gene that may play a role in some of the penetrance of that relationship. On the flip side of that, we have the complex traits, which are typically the result of many, maybe it's hundreds of susceptibility loci or genes across the genome that interact with not only one another, but oftentimes a single or an environmental profile that sort of collectively result in the complex trait of interest. We can study these complex traits and their genetic architecture using genome-wide association studies, where we look at statistical differences in allele frequencies between cases and controls at individual positions across the genome. These are called SNPs. These types of studies have been performed for thousands of phenotypes now, and we see the distribution of those risk loci across the human genome, as well as the X and Y sex chromosomes on the right side of the screen here. The important take home from this image on the right side of the screen is that individual risk loci for various phenotypes are not localized in individual portions of single chromosomes. They are actually spread across the genome, and this is termed polygenicity. We also see that various regions of the genome do not confer risk for one single color data point, meaning they don't confer risk for one single type of phenotype. This is termed pleiotropy. We can evaluate this both sort of polygenicity and pleiotropy with something called genetic correlation, sort of overlap between effect estimates for risk loci for various phenotypes. In the heat map here, we see across the horizontal axis a collection of mental health phenotypes, including some others. And on the y-axis here, we see phenotypes related to sort of cognitive ability or educational capabilities, going down into personality phenotypes, some externalizing behaviors, and some anthropometric phenotypes. The traits that are most interesting for the context of this presentation today are those highlighted in the red box. We'll be looking at relationships between mental health and those education-related phenotypes on the right side, or on the left side of the screen. So what we don't yet understand about the relationships between education and some of these mental health outcomes is how the pervasive effects of education, and we're also looking at socioeconomic status, influence what we can learn from the GWAS of those genetically correlated mental health outcomes. We're also interested in the reverse relationship as well. What is the sort of specific architecture of education and socioeconomic status that is independent of some of these mental health outcomes? So to do this, we adjust all of our GWAS of interest for the per SNP effects of other related GWAS using multi-trait conditioning and joint analysis, or MT-COJO. A diagram of what our analysis pipeline looks like is shown here, where we have a collection of mental health outcomes, and we look at trait relationships using those original GWAS for each of those mental health phenotypes. Then we condition those GWAS using education phenotypes, and we look at the same trait relationships. Then we condition those GWAS with socioeconomic status, and we once again look at trait relationships. And then we compare all of those back to what the original relationships looked like. So I'll start by showing some data about latent causal variable analyses. And this is essentially a way at looking for putatively causal relationships between two traits using SNP information for the genome-wide association studies. So in the sort of pie graph on the right side of the screen, in gray data points, we see the trait relationships from LCV using the original GWAS. And we see one trait pair that survives multiple testing correction, and that is the left subcallosal cortex volume and obsessive compulsive disorder. As we go around that pie graph, we see various colored data points that are all of the same phenotype relationships we looked at in the gray bar or the gray segment, but now both phenotypes in that pair have been conditioned for an individual education or socioeconomic status phenotype. And what we see is this abundance of putatively causal trait pair or trait relationships that are coming out of these conditioned experiments that were not detected in the original unconditioned GWAS. So if we look at sort of a map of these relationships, we see that there are 4 volumetric measures of the brain that sort of mediate a lot of these relationships that we see here, where the colored arrows indicate the direction of effect determined by LCV, and then the correlation, or the genetic correlation, sorry, between those phenotypes is shown above each of those colored arrows. So for example, we see on the top here a decrease in extroversion by several conditioning experiments is putatively causal for a decreased left subcallosal cortex volume. And that's kind of how we would read the rest of this, this map as well. Next, we perform structural equation modeling, again, using the same process where we look at the structural equation models using the original phenotypes, and then the same models using the conditioned version of each of these phenotypes. I want to point out 2 major findings from this SEM experiment. The first of those is after we condition with the math-related phenotypes, self-rated math ability and highest math class, we see that major depressive disorder and neuroticism are no longer loading maximally onto the same factor. And these, if we start looking at the patterns in those factors, we tend to see emerging some anxiety tension factors and a worry vulnerability factor, sort of representing those 2 relationships. Next that I want to talk about is income, and we see that in our figure here. Income is the only phenotype where after conditioning, our GWAS of mental health outcomes no longer converge on a 2-factor model. We actually converge on a single-factor model. And this is quite interesting because in Mendelian randomization studies, we oftentimes recognize, both my lab and others, that income is a major mediating phenotype in some of these causal estimates that we're detecting by MR. And finally, I want to discuss some of the cell type mediators that we believe are sort of causing these relationships that we're detecting, or at least responsible for some of the relationship. I'm showing today major depressive disorder, and that's seen on the right side of the screen. We have the original GWAS. These are outputs from FUMA, the cell type enrichments. So we see a number of transcriptomic profiles for cell types that we're actually able to differentiate, including the gestational week 10 stem cells and GABAergic stem cells, for example. Or, I'm sorry, gestational week 10 stem cells and GABAergic cells. When we condition on the self-rated math ability phenotype, we start to see this different pattern of differentiated cell types transcriptomic profiles, where we, for example, start to see independent associations with the inhibitory neuron 5 and the general class of neurons here. We also perform these analyses with bipolar disorder and schizophrenia. Those figures are not shown here, but we do identify the differentiated transcriptomic profiles for gestational week 10 stem cells and gestational week 26 GABAergic neurons. So to summarize and conclude, by adjusting for the effects of SNPs on education and socioeconomic status, we're able to uncover novel genetic correlations and novel putatively causal relationships between brain imaging, personality, and psychiatric or mental health phenotypes in general. We're also able to identify cell types and prioritize some of these cell types moving forward, hopefully closing some of the gap between GWAS findings and some translatable findings. And finally, education and socioeconomic status, as we see here, may mask some of these underlying biology findings. But what's really important is that all of the findings we report today could not be discovered without considering the effects of several education and several socioeconomic status phenotypes. And with that, I will conclude by thanking our funding sources, as well as the Polamonti Lab and my mentor, Renato Polamonti at Yale. So thank you, everyone. And that is all I have.